Hello, everybody. You are listening to Through Time and Clades. My name is Albert,
I'm Joan.
and we are back with another one of our monthly news episodes. We'll be talking about some new scientific studies that caught our eye in February of 2024, the year that we're recording this. So, um, before we dive into the stories, as usual, how have you been doing?
Pretty okay. I've been busy with a lot of different things.
Mm,
Um, the biggest thing is that I finally have a job now.
congrats.
Um, a little thing on the side that I can do to make some extra money, um, to help support, me and my ventures. And I'm very much looking forward to it. It's at an antique store. I'm going to be helping out with the online marketplace and like kind of keeping track of items for sale.
Hmm.
And it's kind of fun because I like antique stores, vintage places, thrift stores. If I had a nickel for every fun thing that I found in a thrift store, I'd be able to buy out the thrift store. It's one of those things. Um, but it's going to be fun, and it has a really nice schedule, so that way I can continue to work on the show as well.
That's good.
So uh, I'm, knock on wood, there shouldn't be anything that should not interfere with our regular recording schedule anyway, even
Mm,
though it tends to be monthly. Especially since we got some exciting things coming very soon.
mm,
Um, we'll talk about that at the end of our episode. Um, Otherwise, I've been very good. I know the last time we recorded, I brought up the fact that on February 20th, this past February 20th now, was going to be 200 years of dinosaurs.
mm,
200 years since Megalosaurus and it was described scientifically, and thus the dinosaurs were first described scientifically. Um, and I was dog sitting that time, but I was able to celebrate in my own way. I had a huge movie marathon basically,
mm.
where I grabbed a bunch of dinosaur and paleo films from my youth, and series, and I just kind of watched them all again. And oh gosh, that was an adventure. What did I do? I It was T Rex Back to the Cretaceous, which was an IMAX film back when IMAX was new, and their gimmick was 3D. I watched Disney's Dinosaur. I watched both Walking with Dinosaurs, the original series,
Mm.
and When Dinosaurs Roamed America, because I'm fond of them both.
Yeah.
Uh, I watched Jurassic Park and then Jurassic Park 3 because I have a soft spot for Jurassic Park 3.
Mm.
That's a whole story for another time. Um, and yeah, really, that was about it. I just of course, I also watched some Your Dinosaurs Are Wrong and
Oh,
some Rick
nice.
Raptor stuff because that's just to fill out in between the films. Um, it's very wonderful. It really makes me think. I've loved dinosaurs for as long as I can remember, which is really my whole life, and it's just amazing, to reflect on these animals and how much we've learned in that time.
Yeah.
Uh, it's incredible, like not even just the animals themselves, but the kinds of questions that we can actually answer now are vastly different from just 100 years ago,
Yeah.
just 50 years ago, really. It's um,
Mm-hmm.
it's just amazing. I get nostalgic and emotional about it. Um, it's Yeah, it's just been a crazy time these last couple of weeks. Um, Really sad, unfortunate news. Oh, I was devastated when I heard this. So last Thursday, as of this time of recording, we lost Franz de Waal,
Yeah.
who was a primatologist, an animal behavior scientist, an ethologist, um, who really did a lot of formative research on chimpanzee and other primate social behaviors that helped dispel the then widespread assumption that primate societies were inherently violent, that they were frequently led by alpha males who bullied their way around, often with brutal violence, to establish their places. When in actual fact, so-called chimpanzee politics, to echo one of his books, is very nuanced, and often there's a lot of males, working with each other and settling disputes without resorting to violence. And so he was very formative in a lot of that research. And I mean, he's written many wonderful books that I highly recommend.
Mm.
There's two in particular that I know we talked about when we interviewed Meg recently. Um, are we smart enough to know how smart animals are? and then Mama's Last Hug,
Yeah.
which are two books that kind of they go well together. Um, the Mama's Last Hug talks a lot about like empathy and are we smart enough to know how smart animals are? Is basically can humans objectively know about animal behavior, um, and how much can we know with our limited senses? And he was just a very nice guy. All the interviews that I've seen with him, he was a good science communicator. He could clearly tell you his research, and you would understand it immediately. Had a great sense of humor, and he died relatively young too. It's
Mhm.
crazy. Um, and so I just wanted to give definitely a shout out to him. Uh, that's always sad news.
Yeah.
Um, these sorts of things are always least expected. You going through your day, and the next thing you know, oh, this person you really like has died.
Yeah.
Like shit. Okay. I guess I got to live with that now. Um, but otherwise, things have been pretty okay for me. How about you?
Yeah, I would say things are pretty okay for me too. Um, yeah, I don't have too much unusual to report, I guess. Mm, various projects at various stages of completion, and I'll wait until the time is right to discuss them. But I think they're coming along okay. And yeah, I heard the unfortunate news about Franz de Waals as well, and I haven't read as much of his work as you have, but I was definitely familiar with what he had done, and definitely a very influential researcher in his field. Um, he will be missed and mourned, I'm sure. On a lighter note, we did find some exciting stories to talk about for this month, haven't we?
Yeah, that's true. We kind of did a funny thing again with our story selection,
Yeah.
where each of us kind of gravitated towards the same topic for each of our stories.
Right.
So, Al, you picked stories about fish, about ray fin fishes,
Oddly enough, yes.
um, and I picked stories based on plants, flowering plants, and so that's kind of what we're going to stick with this time around. Um, but before we do that, we do have some more follow up to the follow up that we had the last time that we recorded for our January episode.
Yeah.
So, if we may, let's jump over to the next slide. Um, so, in that last episode, being our January 2024 news episode, we discussed ancient DNA studies on North American jaguars that seem to clarify that the giant Pleistocene jaguars, previously thought to be either a subspecies or a separate species, were actually just a subpopulation of jaguars that became specialized towards giant size. So they are within the variation of living jaguars today. Um, in that discussion, I had wondered aloud whether any similar studies had been done on the various extinct Ice Age lions that have been very well studied, and whether they were subpopulations or subspecies of lions, or instead closely related species to lions, or something else entirely. Um, so, of interest in this case is the Eurasian cave lion and the American lion. These are two kinds of big cats that have been allied to the living lions, but were much larger and stockier animals. So the former ranged, as the name suggests, throughout Europe and Siberia, and even into Alaska. And it even features on cave art by Paleolithic peoples, which seems to indicate that the males lacked the big manes of living lions. And of course, we have permafrost specimens too, mainly of cubs, that show that they had thick fur and counter shading on their bodies. Now, the latter lived primarily in North America, south of the ice sheets, at least as far as Mesoamerica. There are claims for South American lions, but those are heavily disputed.
Hmm.
And this was even larger than the Eurasian cats. But it's unclear how similar this species was to them or to the living lions in coloration, fur patterns. But all the paleo art that I've seen usually depicts them without manes either.
Hmm.
Not always, but sometimes. So that's still an open question. Well, it turns out that in preparation for this episode, I did some digging, and sure enough, we did have ancient DNA studies
Hmm.
on these cats, and a fair bit of them at least. Of interest, I found was a collection of papers from 2009, 2016, and 2020, variously authored by Ross Barnett, Beth Shapiro, Mark De Manuel, and many other colleagues. Their names often appeared multiple times in these papers, so it's like you're seeing research conducted over time throughout these papers, which is kind of fun.
Hmm.
Um, so throughout these papers, they sequence DNA from both the extinct and the living groups of lions, and from a very wide geographic range, good sample sizes. And they all pretty much came to the same conclusion. So, the Eurasian cave lion and the American lion are not true lions after all. That is, they don't belong within Panthera leo, which is the lion species. That itself was consistently shown to be a distinct lineage separate from a sister group that included the extinct lions. Uh, so you have a common ancestor. One branch goes off to form the living lions of today, and then a separate branch goes off and then splits to become the Eurasian cave lion and the American lion. So nowadays, those two are best understood as distinct species. So the cave lion would be Panthera spilea, and the American lion would be Panthera atrox. There was no strong evidence of ancient admixture with any of these panthers, and their lineages had seemed to have been separated for quite some time. So that common ancestor of the living lions and the extinct lions split off around 500,000 years ago, and the American lion and Eurasian cave lions would have been genetically isolated by 340,000 years ago. Despite no significant barriers between the two, there doesn't seem to have been any interbreeding as well between the American or the Eurasian species. The living subspecies of lions, so the ones that are found in Asia and North Africa, and then the ones in Eastern and Southern Africa, which are both their own subspecies, they became genetically isolated and separated around 70,000 years ago. Although with those, we do have clear evidence of gene flow between them. There was one really fascinating result that I didn't know about from the 2009 study with the um, cave lions in Eurasia, along with several other megafauna in its ecosystem that it coexisted with, that shows that they underwent some pretty serious genetic bottlenecks after 48,000 years ago, which just so happens to be the time when the Eurasian dispersals of Homo sapiens started to become a lot more widespread.
Hmm.
Um, and so this suggested to the team that the changing climate and environment in the last interstitial may have affected their numbers initially and left many of these species vulnerable to the later megafaunal hunting that would have ultimately done them in. So that I didn't know about that. That was fascinating to
Hmm.
discover. But yeah, there we have it. So as far as all this latest research can tell us, the Eurasian cave lion and the American lion are best thought of as, I'd suppose, stem lions
Right.
that
Yeah.
did not descend from the common ancestor of the living lions. So we shouldn't be calling them subspecies of Panthera leo anymore.
Hmm.
Both of them are distinct species in their own right. So that clarifies that. Do you have any comments on this?
Not too much, but I'm glad you figured out the answer to your question. I did remember that there had been studies, ancient DNA studies on these forms, but I did not trust my memory enough to remember what the actual results were. So, and I know that there has been a lot of debate about the taxonomy of these taxa historically. So yeah, no, it's good to get this clarification.
Absolutely. And I do apologize in advance for this slide. I should have labeled these properly. On the left is an American lion
Mm,
skeleton,
right. Yeah.
and on the right is the Eurasian cave lion skeleton. Um, but if you're familiar with big cat skeletal anatomy, you'll see clearly that how stocky these animals are and
Mm-hmm.
how kind of massive their proportions are compared to skeletons of lions, so to speak. But yeah, I mean, obviously the common names are stuck with us now. I don't see any reason why we shouldn't be calling these American lions and
Hmm.
Eurasian cave lions anymore, even though they're not true lions in that sense.
Right.
Um, I guess your mileage may vary. I mean, we call pumas mountain lions, but
Right.
they're clearly not lions.
Yeah.
So, and sometimes they're called American lions too, which is kind of weird.
Oh, that's
Um,
odd. Yeah.
yeah. So I guess I mean as long as you understand that these are not Panthera leo, I don't think it's a big deal.
Yeah.
Um, but yeah, that's our follow up. So now let's jump into our regular news stories. So I'm going to lead us first with my first plant story. So let's
Mhm.
jump to the next slide. Um, yeah, so my first plant story is another of these large scale phylogenetic studies of evolutionary and biogeographic history. In this case, this is by Oscar A. Perez Escobar and colleagues. This team has decided to set their sights on the orchids, or Orchidaceae, which are themselves a fascinating clade of flowering plants. So let's spend a little time with them to kind of reintroduce them to our audience. So there are over 29,500 species and 736 genera of orchids. They are spread out across almost all known regions of the world. They're mainly associated with tropical rainforests,
Mhm.
but orchids have also adapted to temperate and even polar areas. So there are species that live in Alaska and Greenland and Siberia, of all places, even spreading above the tree line of non glaciated mountains. Although extreme deserts and just absolutely frigid, icy lands lack orchids for reasons that will become obvious very soon. So, orchids can be considered one of the most specialized groups of flowering plants, just in terms of their relationships with their insect pollinators. In many cases, they have formed a relationship with one species or even one sex of one species of
Mhm.
insect,
Yeah.
as in the case of orchids that have adapted their flowers to resemble the females of certain wasp species in both appearance and fragrance. So they trick the males to mate and then fertilize their flowers. Um, there's one cool species, Darwin's orchid,
Yeah.
that has such lengthy flowers that one species of hawk moth has evolved an equally lengthy proboscis to reach its nectar. And in fact, when the Darwin's orchid was first discovered by, Charles Darwin,
Mhm.
the moth was not known.
Yeah. Mm.
But Darwin was like, well, there has to be something out there that's pollinating this thing, otherwise this thing wouldn't be here. Um, and I think it was like 100 years later or something that they eventually found the moth after all. So
Yeah.
that was a long time mystery in biology for a time. Um, there are some orchids, like the genus Corallorhiza, that are parasitic. They have no leaves, they have no roots, and in some cases, they don't even have chlorophyll uh, in their cells. And they completely utilize rhizomal fungi to provide all of their nutrients. And one of these orchids even lives underground. So this is a Rhizanthella gardneri, which is
Mm.
found in Western Australia. It grows entirely below the soil and is pollinated by termites, gnats, and humpbacked flies. It's a very bizarre species. And that's, of course, to say nothing about the variety of orchids whose flowers have just the craziest range of shapes. You can Google some of these right now. Google weird orchids, and you'll get everything, ranging from the cute Dracula simia, which has like these little monkey faces that their flowers make. Um, there's a very elegant species, Pectolysis radiata, that resembles an egret in flight.
Mm.
And there's even a spoopy orchid, Dendrophylax lindeni, which looks like little nighttime spirits that float around, like classic ghosts. Really fascinating. Now, among the angiosperms, what distinguishes the orchids is that their flowers are almost always bilaterally symmetrical, rather than the radial shapes that we're all used to when we look at wildflowers. And they also tend to have their stamens fused to their pistil. So the male and the female reproductive organs have become one structure,
Mm.
and that makes the transfer of pollen much easier to the pollinators, often in a little sticky mass of pollen. Now, orchid seeds are also stupidly tiny, and they are released in these little special capsules that can hold up to like 1 million seeds,
Hmm.
which are then released and spread in the wind. Now, however, the guarantee of new growth for orchids does rely on fungi. You remember the parasitic orchids I just mentioned. Well, orchids are almost always associated with mycorrhizal fungi, which provide the moist and nutritious soil conditions that encourage the growth of new orchids. So this is why cultivation of these plants can be tricky if you're not careful. And orchids have certainly been cultivated widely by people. They're popular ornamental plants. I mean, they look pretty. They're often small enough that you can keep a bunch of them in one place. And some species are even important food crops. So, if you love vanilla, and
Hmm.
I certainly love vanilla, you can thank an orchid. Usually it's Vanilla planifolia, but there's other species that produce vanilla. Mesoamericans, since at least the 1100s AD, have harvested and fermented vanilla for beverages and food and perfumes. And we now have evidence that other peoples cultivated other species of vanilla. In the land of Canaan in the Bronze Age, so about 3,600 years ago, there was recent archaeological evidence that found that people were growing vanilla there too, um, which is a technique that was then subsequently lost over time. So clearly, humans and orchids have a pretty close relationship, but fossils of orchids date back only as far as the early Eocene epoch of between 55 and 40 million years ago. And in particular, that's within Baltic amber. So we have pollen attached to a fly, and that's our oldest orchid fossil evidence. But of course, molecular clock studies, as tricky as they can be, have argued for an origin and subsequent radiation in the Cretaceous period, anywhere from 120 to 76 million years ago, at least. So, kind of hugging the boundary between the early and the late Cretaceous. Not too long from the origin of flowering plants themselves, which is interesting. Now, there has been some question regarding the origin of the orchid clade and comparisons with their sister groups within a larger clade called Asparagales, which, yes, includes the asparagus, but also things like onions and aloe and irises. That has suggested that orchids evolved in an Australian birthplace. And as that continent was still part of Gondwana at the time, some attention has been drawn then to Antarctica and South America as well, in terms of tracing their radiations. And of course, given the vast distribution of orchids in almost all regions of the world, it has been wondered as well how did orchids diversify and spread out across the continents as they have? Because they're everywhere. So, Perez Escobar and colleagues have decided to do the hard work and investigate these questions. They combined genetic information from a number of botanical databases, and they sampled 448 species of orchids, consisting of 38% of all genera, or about 285, across the majority of the higher taxa of orchids. The DNA sequences were then associated with another data set that was based on biography and included 2,320 orchid species arranged by the modern biogeographic realms um, for their origin, because these could then be backtracked through geologic data and that displays the movement of the continents from the Cretaceous period onwards. So you can actually track possible dispersals at that time. And this was then associated, and this is bananas to me. This is associated with another geographic database with every single orchid species described, 29,524 in all, and collecting, in some cases, up to 1,000 locality records for each species. And lastly, the team examined the patterns of species richness using this data through calculations of the average number of species by country per grids of about 100 kilometers by 100 kilometers. So clearly, the team went above and beyond to survey as much of the orchid world as they could to create the best results possible, and I certainly applaud that. The only unfortunate thing was that the team did not use fossil calibrations for their massive phylogeny, even though fossils do exist that are traceable to known subclades. We can pin them down pretty clearly. But I mean, given everything else that they've done, I think we can easily forgive that. So the consensus tree is shown at the left of this slide. Though the main purpose of this tree was to analyze the origin and spread of the group, there were some neat discoveries in orchid phylogeny that the team were able to make, if you're interested in this sort of thing. Mainly in placing a few species and genera in new places. So, like, there was one species called Cooktownia robertsii, which is monotypic, which is it's the only orchid of its type. It's found across the western Indian Ocean islands. What was found to be a sister species to a genus called Habenaria, the rain orchids, which is completely global in distribution. So, little results like that. But, we're focused on the big results here, so let's jump into those. These have basically shifted the discussion of orchid origins in an almost unexpected direction. Rather than a Gondwanan and Australian origin for the clade, the study found it most likely that orchids evolved on Laurasia, in present-day Central America.
Hmm.
The team discovered that not only did Central America, and especially the area of Costa Rica, have one of the highest species richness areas for orchids in the world, and even then that's next to the northern Andes and New Guinea. They also had the most, and I quote, rapidly speciated lineages of orchids. In fact, according to the results, 7 out of 10 of the countries with the most species of orchids were in Central and South America. It seems that those previous results pointing to Australia were due to biases from outgroup sampling.
Hmm.
So remember we talked about the team, previous studies would look at related members of the Asparagales clade that are related to the orchids. Well, it turns out that the previous researchers using those related plant clades were using species with what are known as relictual ranges today. So they're kind of relics of a unique independent dispersal, and thus they're not representative of the range of the larger clades as a whole. And so, thus, the story of orchids, as told in this paper, goes something like this. Following the rise of the angiosperms by the early Cretaceous of 130 million years ago, which is where the molecular clock data puts them, the first orchids had evolved on the continent of Laurasia, which paleobotanists did suspect was a major area of flowering plant evolution, such as, it's the area where yams and palms likely originated. The date has been tentatively placed based on the newer molecular clock estimates at 83 million years ago, plus or minus 10 million years, according to the molecular clock. So that would mean a late Cretaceous origin for orchids. Specifically, this region was in the extreme south of Laurasia, which would one day become Central America. And from there, beginning in the Paleogene period, the orchid lineages began to spread out across North America and into Laurasia. And many of these eventually survived only in the tropics of Southeast Asia today. So there's broken chains in this ancestry here. Most of the globe began to experience cooling through the formation of the Antarctic ice sheet between 35 and 10 million years ago, and this led to dispersals from Southeast Asia and into Australia and Africa from there. Some orchid groups even went back into the Americas
Hmm.
via Eurasia. There's a clade called Cymbidiae, for example, that did just this. What is especially surprising is that despite the nearly 30,000 species of orchids alive today, most of that diversity seems to have evolved only within the last 5 million years during the Pliocene and Pleistocene epochs. Much of this in the neotropics happened through the formation of the first cloud forests and hilly grasslands at about the same time, to have created very numerous and prime microhabitats for orchids to then settle in and diversify from there. So, is this the last word on orchid evolution? Probably not. But I mean, this is certainly an impressive study that is going to set a standard for future research. And honestly, even without the fossil calibrations, the team was able to basically confirm the presence of orchids in times and places that match exactly with the known fossil specimens that we have.
Hmm.
So I think that's a pretty good sign. And I mean, to me, it's a fascinating example of what is a sometimes apparent mismatch with a clade's diversity and its history. we talked about this with cycads in a previous episode, where cycads are a very old lineage of plant, but most of the living cycads only evolved recently.
Hmm.
Well, here we go again with orchids. They've been around for at least 83 million years, and yet the majority of their current diversity only originated within the last 5 million years.
Wow.
And that's just wild to me. Uh, so I'd love to get your thoughts on this paper.
Yeah, as always, I really enjoy this type of paper, as you know. And orchids are one of those groups that are, yeah, like you said, just really fascinating. Because I think if you have any interest at all in evolutionary biology, even if you don't know much about plants, which I've made no secret that I don't really, um, you're bound to come across orchids eventually. Well, in part because of all their fascinating ecological and evolutionary relationships with species that they depend upon, or codepend upon rather. So they're pollinators, and the fungi, like you mentioned. They're definitely an amazing group of plants, and it's not too difficult to appreciate that massive diversity and the amazing adaptations that they have. And it is really interesting that they found such different biogeographic results from previous studies. But yeah, I think if it's because of the outgroup selection, that would probably make a lot of sense. And we have discussed before on the show how outgroups can influence the results of your phylogenetic analyses. So I could definitely buy that. And yeah, the fact that they apparently didn't use fossil calibrations is a bit of a shame. I'll have to probably read the paper myself to figure out um, exactly what methods they used. But um, the fact that they did predict correctly where fossil orchids could be found, um, I would agree, seems like a good sign. Um, and so, yeah, I think it's great that we have this new study shining this new light on orchid evolution. They're definitely a group of organisms that really deserves that kind of attention, really.
Absolutely. And I mean, even going back to the whole most orchids alive today only evolved within the last 5 million years.
Yeah.
That's simply because of the changing rainforest conditions that are established today. I think it really underscores another myth in biology that the so-called jungle, right, the
Mm.
tropical
Oh,
rainforests
yeah.
are supposed to be these ancient, unchanged places
Hmm.
for millennia,
Right.
when clearly, when you factor in the glaciations in the Pleistocene, most rainforests of today are relatively young places,
Yeah.
um, because often many of them have reclaimed land that was originally, grassland, when the climate was, cool and dry
Hmm.
in the glacial periods. And so many of these rainforests were like expanding and then contracting multiple times. Um, and really, the tropical rainforest as an environment, or as like a biome that exists, we think about, a large, wet, rainy area dominated by um, angiosperms and angiosperm trees. That's a Cenozoic development.
Right.
Um, there were no rainforests in the Mesozoic. The image of dinosaurs in Jurassic Park, hiding out in the dense, thick foliage. Um, that was not a thing in the Mesozoic. That's a Cenozoic development, and it really owes a lot to the evolution of flowering plants and their pollinating insects, and the global environmental conditions. Because when the Cenozoic started, they underwent extreme hot phases,
Hmm.
and there were jungles everywhere, even in the Arctic. You could find alligators and palm trees in Greenland, and we have. Um, and of course, over time, as the globe has slowly cooled, all those rainforests have shrunk and were replaced by taiga or temperate forests, and their distribution today is the subsequent result of that. This little equatorial band of forests that we have, and even then, those regions have been lived in by people and maintained by people for a long time. So we've talked a lot on the show about the Amazonians and how much of the rainforest was essentially densely populated, and then following the population collapses through various factors, people moving or colonialism, those forests have reclaimed those cities. So even then, in those parts of the Amazon, the rainforest is only like 500 years old.
Mhm.
Um, it's it really kind of skews your perspectives. But yeah, definitely, if you thought that rainforests were old, and like they kind of are, that's a bit of a wake-up call, you know?
Yeah.
As far as global ecosystems are concerned, rainforests are some of the youngest. I put money on that.
Mhm.
But yeah, this was a great paper, and I'm excited to see new studies like this, as you are as well. And there have been quite a few that we've had to pass up on in this
Right.
show, but they just keep coming, and I love seeing them.
Yep.
You know, we're really fleshing out these clades uh, for future researchers. Um, but I guess now, jumping under the waves and into your fishy stories, Alba, I understand you got some for us. Why don't we start with your first one?
Sure thing, yeah. So, funnily enough, both of the stories, as you mentioned earlier, that I picked for this time, both have to do with fishes, and not quite my usual focus. Yeah, unless you unless you do the usual the old joke that all vertebrates are fishes. Um, but yes, so these are like fishes as we would think of fishes. They're like not as land dwelling vertebrates. So that's quite interesting. But they are incredibly fascinating. I think, as I think the stories I picked this time will show. Um, and so the first story I picked is about how a specific species of fish produces sound, which is, I think, on the surface of it, is going to sound strange to a lot of people because we don't really think of fishes as making sound very much. Like, what does a fish sound like, right? um, well, it turns out that actually a lot of fishes make sound. And so this kind of goes along with a study that we talked about on a previous news episode, where the authors surveyed sound production in a wide range of vertebrate animals, backboned animals. And that study mostly focused on land dwelling vertebrates like us, so the tetrapods, basically. And classically, it is thought that only a few groups of tetrapods evolved the abilities to make noises, to communicate with each other. So groups like frogs and mammals like us, and also birds, a few of the lizard groups are kind of the more well-known sound makers. However, that study showed that a lot of things that we don't normally think of as being very vocal also do make sounds too. So things like turtles, some of the turtles actually make quite a few noises, or even things like salamanders and lungfishes also make noises too. But that study, as I mentioned earlier, mostly focused on land-dwelling vertebrates and their immediate relatives. So to be more precise, it focused on the lobe-finned fishes. So these are the group of fishes that we belong to, you could say, and we are characterized by having kind of limb structures where the limbs are basically supported at the base by a single bone, followed by two bones, and then followed by a lot of bones, which should sound familiar because that's exactly how our limbs, even though they're not fins anymore, are structured. But the group that is most closely related to the lobe-finned fishes that are alive today are a different group. And this group, some of them can come onto land, and we've actually talked about that before too in a previous story. We talked about catfishes that can walk around on land, kind of. But they haven't really kind of colonized the terrestrial realm as much as we lobe-finned fishes have. And this group is called the ray-finned fishes. But lest you think that the ray-finned fishes are not successful, that you couldn't be further from the truth, because they are incredibly diverse. They're at least as diverse as lobe-finned fishes, if not more so, in terms of number of species, even though they're mostly confined to the water. And the fin structures of ray-finned fishes are quite a bit different from the lobe fins. So they also have a single bone supporting the base of their limbs, which in their case are always fins. But radiating out from that single bone are kind of longer and thinner spine-like bones, also known as fin rays, and that's where the name ray fins comes from. And so this is a group of fishes that includes most of the things that we think of as fishes, except for sharks, except for jawless fishes like the hagfishes and lampreys, and except for the few lobe-fin fishes that have stayed fishy, and so in the modern day, those would be the lungfishes and coelacanths. But aside from those things that we just mentioned earlier, pretty much anything you can think of today that you think of as a fish is a ray-fin fish. So think goldfish and salmon and cod and seahorses and pufferfish. All of those are ray-fin fishes. And so, as it turns out, quite a lot of ray-fin fishes make sounds too. Now, the way that they make sounds is often pretty different from the way that we lobe-fins make sound. Two of the most common ways that ray-fins make sound. One is by vibrating an organ called the swim bladder. We'll come back to that in a bit. And the other is by what is called stridulation, which is basically by rubbing two different body parts together. And in the case of ray-fin fishes, they're normally rubbing parts of their fins together. So those fin rays, for example, like some species can rub those together, or maybe like grinding their teeth together. That's also a form of stridulation. In fact, I guess as one example of a well-known group of ray-fin fishes that vocalizes by stridulation, clownfishes actually do this. I think a lot of people don't know this, but yeah, clownfishes actually communicate with each other. They defend their territories by making these grinding sounds with their teeth, basically. And if you ever watch Blue Planet 2, and watch the clownfish sequence in there, you can actually hear these noises that they're making, and yeah, they're using their teeth to make those noises. Now, when you're watching nature documentaries, you do have to be careful when it comes to like animal sounds, because a lot of the sound effects are added in post-production by using artificial means, basically, and so do not actually represent the actual sound that was going on when that footage was filmed. And a lot of that comes down to logistics, because a lot of this stuff is filmed using, say, drones, for example, or filmed from cars and such, and there's going to be the sound of like engines running if you actually included that in the final film, so they have to like reproduce the sounds artificially. But in Blue Planet 2, in the very final episode, they talk about how while they were filming the clownfishes, they noted that the sounds of motorboats passing overhead were disrupting like their communication, and so the creaking noises that the clownfishes were making were like the actual noises that they were making during that scene. So that's pretty cool. So yeah, if you didn't know that fishes make sound, that's one good example of that. Now, the particular species of fish that this new study that we're going to talk about today was focusing on is a species called Danionella cerebrum. And if you've never heard of the species, well, you're not alone, because this species was only described by science in 2021, so it was only described a few years ago. Before that, this was a species that was not known to science. It is a freshwater fish and found in freshwater bodies in Myanmar, and it has a number of kind of interesting aspects to its biology that have been identified. First of all, it is one of the smallest known vertebrates in the world. Its body length is only about 13 millimeters long or less, so it's barely a centimeter long. And it also has the smallest known brain of any vertebrate, so a somewhat dubious honor, but that's probably why it's called a Danionella cerebrum. So cerebrum means brain, so it's named after this notable trait. But something else that's interesting about its biology is that the males of this species are known to make sounds in social contexts. So basically, when they're around other members of their species, they make these noises. Now, I don't think it's well understood like what these noises actually mean. So, are they like defending territories or are trying to attract mates or such? I don't think that's very well studied. But it is known that they make sounds, and these sounds are pretty curious, even for sounds made by ray-fin fishes, because they're made in short pulses. So they're unlike most sounds that would be made using the swim bladder. So most fishes that make sounds using the swim bladder make kind of longer tones when they're basically kind of stretching their swim bladders with specific muscles or vibrating those swim bladders. Whereas Danionella cerebrum, those sounds, they sound like more like short clicks, and they every time they vocalize, they kind of make a rapid series of those clicks, in these bursts of sound. So the short kind of duration of these sounds would seem to suggest stridulation is more likely as a mechanism for making these sounds. But that doesn't seem to be the case either, because these sounds are pretty consistent with each other, pretty regular. Whereas if it's stridulating, we would expect like more variation in how the sounds sound, essentially. And so this new study decided to investigate, well, a little bit about what these sounds were like, but also how these fishes are actually making these sounds. Now, if you have heard of zebrafish, so if you are interested in keeping freshwater aquariums, for example, you might have heard of zebrafish, also called zebra danios. And if you're into reading scientific research about biology, you've probably also heard about zebrafish because they're common laboratory animals. Zebrafish are closely related to Danionella cerebrum, so they're probably the closest relatives to this species that a reasonable number of people have heard of. That being said, if you haven't heard of zebrafish, probably the next closest relatives to Danionella that you might have heard of are things like carp and goldfish, which are a domesticated form of carp. So they're all members of a group called Spinopinnophyllum, and so yeah, these are basically very, very small carp relatives. So the authors of this new paper kind of made some recordings of these sounds and also studied what was going on with these fishes. How are they making these sounds in detail in this new study? So one of the interesting things that they found about the sound that Danionella cerebrum makes is that relative to body size, these are some of the loudest sounds that any vertebrate makes. So these sounds reach over 100 decibels at a distance of 1 meter away. And to be more precise, they reach about 108 decibels at that distance. Now, that's pretty darn loud. So you might want to compare that to other types of animals, right? And that's not as straightforward as you might think, because first of all, sound travels differently in the water versus in the air, and also you need to keep the distance consistent when you're measuring these noises or recording these noises, because a sound is going to sound louder at closer range, right? But the authors of this paper do provide some figures for comparison in cases where other animals have been recorded or estimated at distances of 1 meter, and also kind of corrected for the kind of difference in how sound travels through different media. So on land, the loudest animal on record is the white bellbird, which is a tropical bird from the tropical Americas, which makes this very loud sound that at a distance of 1 meter is about 125 decibels. So it's somewhat louder than this tiny fish. But remember, this is a tiny, tiny fish, so it's going to be smaller than all these other animals we're going to compare it to. The loudest land mammal that's been recorded are certain types of bats, which can reach about 120 decibels. Elephants can also reach about 120 decibels. And for comparison's sake, a typical human shout can reach about 105 decibels. So yeah, even a human shouting, under typical circumstances, is not louder than the sound made by Danionella cerebrum. Allegedly, there are instances where human screaming have reached 129 decibels, but it is unclear how those figures were kind of obtained, or under what circumstances those figures were obtained. So those figures are not considered entirely rigorously reliable. In the water, animals can get a lot louder. Sound travels faster through the water, after all. So the loudest animal of any kind on record is the sperm whale, which can make sounds of 239 decibels. So yeah, that's extremely loud. And the loudest fish on record is a marine fish called the black drum, which can make noises of over 180 decibels. Which it does by a swim bladder vibration. So all those examples are vertebrates, and one of the loudest animals ever on record is actually a type of shrimp, So these are a type of shrimp called the snapping shrimp. They have a enlarged claw that can produce very loud clicking noises. And so these shrimp can produce sound of also over 180 decibels as well. So they're not quite as loud as a sperm whale, but they're still some of the loudest animals on record. And these shrimp are pretty small too. So they can be longer than Danionella cerebrum, but also kind of within the same size range. Probably relative to body size, the loudest sounds of any animal might belong to the snapping shrimp. But in terms of loudest sounds made by any vertebrate relative to body size, I think Danionella cerebrum definitely stands out. So these fishes make these remarkable sounds. How do they do it? Well, the authors of the study used a few different means of investigation to try and figure this out. First of all was that they simply took video footage of these fish, high-speed video footage, and they slowed it down to see what was going on when these fishes were making noises. And as you can see, these fishes have fairly transparent bodies, right? So you can actually see through them even while they're alive and see what's going on inside them, what's going on with the internal organ in the skeletons, for example. And so what they did was they took video footage of these fishes while they were making sound and slowed it down to see what was happening inside them as they were vocalizing. Something else they also did was examine dead specimens of these fishes and actually examine the actual anatomy, what was the structure of the anatomy inside the body. And after making their observations, they were able to come up with a model for how these fishes probably produce sound. And so it turns out when Danionella cerebrum makes sound, the sound production correlates with movement of one specific pair of ribs in the body, as well as what seems to be a specialized cartilage, which strikes the swim bladder and compresses it. So the swim bladder is an organ that I promised I would get back to, and so we're going to talk about it now. The swim bladder is an organ that in rayfin fishes is basically the equivalent to our lungs. And I know I've probably said this before on the show, but I'm going to say it again because I know there are certain sources that should know better that always get this wrong. Our lungs did not evolve from the swim bladder. So that was an idea that had been suggested in the distant past, in the early days of evolutionary research, but we have known for a long time that is probably not the case. Instead, the original version of that organ was used for breathing. So in other words, the original version of that organ in the last common ancestor of both ray-finned and lobe-finned fishes were lungs. And it was only in the ray-fin fishes that they became adapted to be used as a flotation organ, in other words, the swim bladder, instead of being primarily used for breathing. Now, there are still some ray-fin fishes that can use the lungs/swim bladder for breathing, and so they'll come up to the surface and gulp air if there's not enough of it in the water. And in fact, this was probably the reason that lungs first evolved in the first place, in the last common ancestor of both ray fins and lobe fins, because that last common ancestor was not an animal that could move around freely on land, probably, but it might have been able to supplement its breathing by coming up to the surface to gulp air. And it was only later, once the lobe fins had actually moved on to land, that they kind of lost the ability to breathe underwater. We lost our gills. And became kind of full-time air breathers using only the lungs. Whereas ray fins kind of took this in a different direction. So there are still some ray fins today that can gulp air and get oxygen from the air using their lungs. But in the majority of living ray fins, the swim bladder is primarily a flotation organ. So it is still used to kind of hold gases, but instead of being used for breathing, it is instead used to control the position of the fish while it is swimming in the water. And in some ray fins, the connection between the digestive tract and the swim bladder is completely cut off now, so they couldn't come up to gulp air and breathe even if they wanted to. So it becomes a full-time flotation organ. Now, even though they cannot breathe using the kind of cut-off swim bladder, this actually allowed the ray fins to colonize environments where they wouldn't need to come up to the surface for air, such as the deep sea. And so that's quite a fascinating bit of the evolutionary history of fishes, is how they use the swim bladder in these different ways. And in addition to breathing and flotation, as we can see, and as we've already mentioned, some ray fins also use the swim bladder to produce sound. And so basically the take-home message here that I want to get across is that lungs did not evolve from the swim bladder. The swim bladder evolved from lungs. It is literally the other way around. But anyways, it seems that Danionella cerebrum is one of these ray fin fishes that uses the swim bladder to make sound. And it seems to do so not by kind of vibrating the swim bladder with muscles, as occurs in some of the other vocalizing fishes, but instead by striking it with this cartilage. Okay, so how does this even work? It seems that the movement of the cartilage is way too fast to be the direct result of muscles pulling on that cartilage. So there's a limit to how fast muscles can move, and according to the author's observations, that cartilage moves too fast to be pulled by muscles on its own. And so that's why they call this ultra-fast sound production in this species. So if it's not being moved by muscles, what is going on? Well, it turns out what the authors hypothesize is that muscles are used to move that pair of ribs that moves along when the sound is being produced. And what happens is that there are specialized muscles that pull on the ribs, and then the ribs move forward, and as they move forward, they contact that specialized drumming cartilage, and so they hook into that cartilage and pull it forward for a very short distance, but then the cartilage is released. The ribs kind of lose their grip on it, and so the cartilage is kind of slung towards the swim bladder, and then it strikes the swim bladder. And so that's how the sound is produced. And so the ribs are paired, as you might imagine, and so are these muscles that move the ribs, and so are these drumming cartilages. So there's one of these drumming cartilages on each side, and there's only one swim bladder. So unlike our lungs, the swim bladder is not paired. The fish can contract only the muscles on one side to do kind of one-sided drumming of the swim bladder, or they can do double-sided drumming of the swim bladder. And when they do this, they alternate on each side. So they might contract the muscles on the left side first and then the right side. And so they can make this more rapid drumming noise when they do that. And so the authors observed both one-sided and double-sided vocalization mechanisms when they were looking at these fishes vocalizing. So it seems that this is how Danionella cerebrum makes sound. And one more thing that the authors bring up that I think is worth mentioning on the show is that similar anatomical structures have been observed in other species in the genus Danionella. So there are a number of closely related forms all in this genus, and I think they're all found in Southeast Asian freshwater environments. Including one species that is also quite recently described. It was also described in the 21st century, called Danionella Dracula, which is a really fun name. And, well, it actually has a very good reason for being given that name, because the males of that species have these projections on the jaws that look like fangs. And so this is really interesting, too, because as it turns out, cypriniform fishes, so the carp and their kin, including Danionella and the zebrafish and such, they do not have teeth in their mouth. They have lost teeth in their mouth. They do still have teeth inside their throat, which sounds weird to us, but it's actually something a lot of fishes have. But they don't have teeth in the mouth. And so what happens with Danionella Dracula is that bones of the jaw become adapted to look like teeth. And so they have these tooth-like projections in their jaw that the males apparently use for kind of fighting each other in territorial contests. So that's why they're called Danionella Dracula. And so it's kind of in this way they kind of have an interesting parallel to birds, right? Because birds, modern birds at least, also don't have teeth, and sometimes some groups of modern birds have evolved ways of getting back tooth-like structures in their mouth that don't involve re-evolving actual teeth, which they might not be able to do. So like they will modify parts of the beak, for example, to become tooth-like instead of re-evolving teeth. And so something similar has happened in these Danionella Dracula fishes. But Danionella Dracula is also known to produce sounds, much like Danionella cerebrum. Now it produces shorter bursts of sounds. And it has also been observed that it seems to have this specialized cartilage and modified ribs that can move like this. So it's quite likely that these are common features to probably most of the members, if not all of the members of this genus. And so, yeah, it's quite remarkable how these very small fishes can pack so much amazing biology into them. And that's definitely a pretty big reason why I chose this story for one of my news stories. So, yeah, do you have anything to add to this?
Nice to see this as a sort of follow up to some earlier studies that we've talked about regarding sound production in vertebrates.
Yeah.
There were, hypotheses regarding like the previous studies that we've talked about that even if we've been able to track like a common ancestry for sound production in sarcopterygian vertebrates, so the lobe fins and the land vertebrates, tetrapods, those like underlying mechanisms might have even earlier origins. So to see that also in ray fin fishes, even in its own specialized forms as this, where you have the vocalizations in some species, and in other species you have the swim bladder and the ribs working together, like in this fish. That's fascinating to see. It's almost like you're convergently evolving sound,
Yeah.
if I'm making sense.
Mm-hmm.
It's a shame that we can't. There's not a way to play the sound of the fish on here.
Right.
To hear that sound, especially considering one of the loudest sounds relative to body size of any vertebrate. That's friggin'
Yeah.
that's crazy. One of the smallest vertebrates produces the loudest,
Yeah.
some of the loudest sounds. That I was not expecting that, to be honest. But more power to this fish. That's
Yeah.
great. Yeah, that's fascinating. Yeah, and you know what? It's interesting that you mentioned that the fish is see-through,
Yeah.
because I know that's been a big discussion in biology for many years. Um, Obviously, in the past, when you wanted to get inside an animal to understand it, you had to cut it open
Right.
and dissect it. But there are many species like this that the body is see-through, and you can not only see inside the animal, but actually see it as it's alive,
Yeah.
and see the organs working exactly. And you can have studies like this where you can study the source of sound for these fish without having to really cut them open.
Mm-hmm.
It is interesting to consider some of the genetic research that has been done in the last couple of years, where they've been wanting to, or different research teams have been wanting to put, I guess for lack of a better word, the genetic code for having a see-through body in other test animals in order
Yeah.
to study them without having to dissect them. I don't know how I feel about that. It's an interesting concept,
Yeah.
but it does make me wonder. Um, at the very least, like you have more ethical research like this, where you just look at the animal itself because it's already open for you, so to speak.
Right.
Um, Very fascinating. Yeah, this was cool. I'd love to see further research on this and on other fishes, especially to really kind of further open up the book of nature on sound production in fish.
Oh yeah.
Because you're right, like it's not a thing people would usually think about. What sound does a fish make? Oh, blub blub. It's the friggin' bubbles.
Right.
But
Right.
no, like they can make their own sounds with their own organs.
Yeah.
It's and in many different ways too. Gosh, what a time to be alive. That's awesome.
Yeah. And if you want to hear the sounds that this fish makes, we will, of course, be linking to the papers that we discuss, as always. And there are recordings in the paper, so you can go check that out.
Perfect. Well, that works out then. Well, in that case, then, if you don't have anything else you wanted to add, we can move forward now with my second and final plant story
Sounds good.
for today. So, this is more of a traditional paleontological description than a paper describing a process or like the lineage of a clade. So this is by Sanchicha Kumar and colleagues, and they have provided us with new fossils of palms from a very critical period of the Earth's history. So palms are flowering plants. They belong to a clade Aracaceae and includes over 2,600 species across 181 genera. And they inhabit just about every part of the world that's tropical or at least subtropical. So Linnaeus, one of the classic classifiers of life back in the 1700s, he called the palms the princes of the plant world.
Hmm.
And it's honestly not hard to see why. I
Hmm.
mean, many of these species are very regal and aesthetically pleasing in many ways, especially with their fronds, which are a distinguishing feature of the group. So palms can have either palmate or fan-like palms, leaves, that is, or pinnate or feather-like palms or leaves. And these can grow either from a solitary stalk, hence palm trees, for example, or in a cluster, as in palmetto bushes. These distinct fronds make for very recognizable plant fossils. And among the angiosperms, they are actually some of the earliest specimens we have for that entire group, with some dating back 80 million years ago. And some of these fossils are very impressive too. Honestly, one of my favorite plant fossils ever
Hmm.
is the palmetto from the Green River Formation on display at the Smithsonian's National Museum
Yeah.
of Natural History. That's a species called Sabolites powelli that lived between 53 and 48 million years ago, and it's enormous and it's relatively complete, and it was preserved in a way, um, with Green River Formation, river sediments. It was preserved with other organisms around it, so there's like little tiny herring-like fishes scattered about
Hmm.
a fossil. It's just beautiful, and I love seeing it every time I get the chance to go. Um, so in this case, it might seem that the discovery of new fossil palms might not be too exciting because they're so commonplace as far
Hmm.
as plant fossils goes. But hey, that should never be the case for any lineage of life on this planet. Any fossil from any group can hold new and surprising and amazing secrets. And that brings us to the study today and why I picked it. So some species of the pinnate variety of palms have spines on their leaves.
Hmm.
And these are used either as defenses from big herbivores or as supports for those species that are epiphytic, or they climb and they grow over other plants. And they're not necessarily parasitic, but they can be. So we have two new fossils of such leaves that have been found at the Madhya Pradesh in central India. And not only do these span from the late Cretaceous to the Paleocene epoch, but they're also found within the Deakin traps. Like, what? Yeah. So these fossils were uncovered back during the early years of the pandemic, so in 2020, 2021. And the Deakin specimens span the boundary between the Mesozoic and Cenozoic eras. Now, for those not in the know, the Deakin traps are a series of preserved lava flows, some of the most significant in the world, for they record a period of volcanic activity in South Asia, back when it was an island continent, that was utterly enormous. And it would have likely had effects on the global climate to some extent during that time. These lava flows, or more technically, solidified flood basalts, these stretch in total over 500,000 square kilometers
Hmm.
and can reach 2,000 meters thick in some places. Um, so these were some serious eruptions. And while they have been argued in the past to have played some role in the end Cretaceous mass extinction event, it is seeming that nowadays paleontologists would agree that the Deacon Traps eruptions had a far more minimal impact on organisms than, say, the Chicxulub bolide
Hmm. Yeah.
that struck the Earth, and which seems to have done most of the damage to life on the planet. In the cases of our palms, these were uncovered within what's known as the mandala lobe of the Deacon Traps, and that spans only 900 meters of 29 intermittent flows across a span of time less than a million years. So between these periodic few thousand year eruption periods, the landscape would recover, and overall it would have been composed of mostly lakes and rivers supporting abundant plant and animal life, including fruiting trees, and palms seem to have been particularly prominent members of the overall flora. Now, the two palm leaves, as mentioned before, preserve evidence of spines, and they represent a newly described species and genus. So, we are introducing Spinopinnophyllum acanthorhynchus. So, the palm with the spiny pinnate leaves. So, this is one of those cases where the scientific name is literal, which I think should be the case all the time, but that's a discourse for another day. So as you can see in one of the fossils here on the left of the slide, as well as the accompanying life restorations, the spines are arranged along the stem, or rachis, of the leaves, in between the leaf segments, which also gives it the species name, which means spines on the rachis. The fossils preserve just the middle portion of the leaves, and they measure between 9.2 and 14.4 centimeters long by 1.3 to 6.9 centimeters wide. As far as the wider classification goes, it was difficult for the researchers to tell which palm Spinopinnophyllum is most closely related to, as multiple clades of palms have spines and are not necessarily relatives of each other. So one of these examples of these living spiny palms is a species called Astrocarium murumuru, which is shown also on the left. Now, that said, in comparing the size and shape of the spines of the fossil species with the living groups, it does seem that the closest match are the spiny palms of a clade called Pactridinae, which is found only in the neotropics, and they typically grow as trees with the solitary branching conditions. These are palm trees proper. Despite the spines, many of these palms have been domesticated and cultivated by indigenous groups going back into pre-Columbian times, especially for the fruit, which is highly valued. Now, as mentioned before, there are multiple uses for spines among palms, but among these species that most closely resemble our new fossils, the Bactridae, these spines are used for protection against large herbivores. And so the scientists have reasonably speculated that the presence of these spines on this genus, Spinopinnophyllum, may have actually been a deterrent for some of the last of the giant dinosaurs, which would have certainly enjoyed their fruits. There's a long discussion about whether herbivorous dinosaurs invented fruits in angiosperms. Of course, again, that's another topic for another day. Natural history can go all kinds of directions. That's just how it is. So while there are a few other spiny palms in the fossil record, these are mainly known from the Cenozoic era, from the UK to America to the South Pacific. So Spinopinnophyllum is technically our first Mesozoic spiny palm. It's the oldest known, and it dates from the very latest Cretaceous at 66 million years ago. Now, its locality in South Asia lends support to the idea that at least the pinnate palms did originate on that landmass and then likely migrated from there onto the greater Eurasian landmass and beyond. So this is what's known as the out of India hypothesis, which is another contrast to the Laurasian model that some paleobotanists said used for the origin of palms. There's some discourse there as well. Granted, this is not really addressed in the paper all that much, but to me it did feel fascinating to see that these fossils represent a species that we know survived through the KPG mass extinction event. The fossils persist in layers at least to 65 million years ago in the Danionella age of the Paleocene epoch. Now, admittedly, this is not so unusual a thing. There are many species and genera that are known from across that boundary. So there's Nothofagus, which is the southern beech tree. That's known from several sites, having made it through the catastrophe. There's many genera of mollusks that are around today, Arca, the arca clam, Fusinus, the tulip snail, Pinna, the pen shell, and Anomia, the jingle shell. These are all known from across the KPG boundary. And of course, those are just examples of plants and animals that are alive today. We have now extinct species as well that made it through. There's a crocodilomorph, Borealisuchus. There are multi-tuberculate mammals, Mesodma and Cimexomes. And then there's Albinurpaton, which is a salamander-like lysamphibian, which belongs to a clade, the Albinurpaton today, that managed to last until the start of the last ice age, just 2 million years ago. So we almost missed these guys as among the living amphibians of the world, which sometimes I stay up at night thinking about that.
Hmm.
So now, these are literally organisms that witnessed the bolide impact and essentially lived to tell the tale.
Hmm.
They made it through the resulting chaos. Now, we know from recent research at several sites that freshwater ecosystems seem to have made it through, or at least recovered surprisingly well
Hmm.
through the extinction event, to the point where, and we talked about this in a previous study, in the thousands of years later, there were giant garfish and turtles and crocodilomorphs that were present right afterwards and apparently thriving and diversifying. So that these spiny palms seem to have uh, grown in a moist wetland environment, as the geologic record suggests, well, here we might have yet another example of a freshwater living species that made it through the catastrophe. And in this case, also seemed to survive the volcanic environment of the Deakin Traps, which must certainly have periodically burned down so many forests.
Hmm.
So it's almost a miracle that this thing survived that at all. Um, and so I certainly anticipate further fossil discoveries from this part of the world, especially because I really would like to know just how organisms would have adapted to a place
Yeah.
as that. Um, Remarkable little fossil palm. Um, and so I just had to share this with everybody. It's a really neat paper, and it's also a nice example of a paper that we don't usually cover on this show. Just a standard description of a new fossil, um, where the authors go in depth to all the anatomical features and comparing it with other fossils and other living groups to show why it's distinct. That's really like the basis of the field.
Yeah.
If you think about it, it's always easy to forget that when we see the bright, flashy, big phylogenetic evolutionary surveys of groups, or that sort of thing. Not that there's anything wrong with those, of course. Certainly not. It's just it's familiar territory to me. So I thought this was pretty fascinating. What do you think?
Oh yeah. I'll definitely have to echo your description of the importance of this type of study because, yeah, those big phylogenetic studies that we love to talk about so much, uh, these scientific descriptions of the individual organisms are the foundation of those studies. Like we wouldn't know about these organisms or what they were like, and we wouldn't be able to include them in those analyses if it weren't for descriptions of new specimens and new species. And so this type of research is very important. And this is definitely a fascinating example of that. I mean, you've already brought up many of the most interesting points, I think, but something that comes to mind is the mention of this form having spines as a potential defense against herbivores, because that's definitely something I've thought about when it comes to Mesozoic ecosystems, because herbivores then were pretty different from the big herbivores that we have now, right? Most of the big herbivores that we have now are mammals, and it is well known that thorns are a very common defense that modern plants have against mammalian herbivores, which makes a lot of sense because mammalian herbivores tend to approach their quote unquote prey, plants that is, with relatively sensitive lips and tongues that wouldn't like to come in contact with thorns very much. Um, but it is also known that in modern ecosystems where birds are the primary herbivores, so think New Zealand, which until recently had the giant moa as the main big herbivores, or Madagascar, where they had until recently elephant birds, um, the plants there don't seem to be as thorny as plants in other types of ecosystems. Um, and instead have other ways of defending themselves. Which we've kind of talked about that before on the show as well, I believe. So I think we mentioned, for example, maybe in the Paleonath episode of Dinosaurs, the second chapter, where some New Zealand plants and some Malagasy plants, they have a kind of very springy structures to their tissues, so it's very difficult to pull pieces of them off, basically. And this matches how large birds would be feeding. So they would be like grabbing like a few leaves or individual leaves and kind of yanking them back to try and pull them off the plants. But if the plants are really springy, it's a lot more difficult for a big bird to do. Whereas if these plants had thorns, that might not be so effective against birds because, of course, birds feed with their beaks, which are a lot harder and probably can be more easily protected from thorny defensive structures. And so you have to wonder, how did plants during the Mesozoic deal with gigantic non-bird dinosaur herbivores? And many herbivorous dinosaurs also had beaks, like modern birds do. So you would expect that thorns would not be quite as common, perhaps. But it is quite interesting because the main big herbivorous dinosaurs we know of from India at the end of the Cretaceous, from the time and place where these fossils were found, were sauropods, which there's actually a little bit of debate about this, but in general, we tend to think that they probably didn't have beaks, at least not in the way that, say, the bird-hipped dinosaurs did, or a lot of the plant-eating theropods did. And so I wonder, if this is the case, maybe thorns were actually an effective defense against sauropods, or at least enough that it would be selected for in these plants. So that's really fascinating to think about. And maybe that's a line of evidence against sauropods having beaks. Who knows? I wouldn't use that as a slam dunk, but it does make me wonder. And the fact that we find it across the boundary, that too is really cool. And like you said, it's amazing, especially considering that it's not only survived the bolide impact, but also coexisted with the Deccan traps at the same time. Truth be told, most of our knowledge of how organisms kind of responded to the end Cretaceous extinction comes from sites in North America. And so to have more fossils from the right time in South Asia instead is definitely a kind of window into a region from this time that we don't get quite as often and don't understand as well. So there are definitely many, many reasons to celebrate the discovery of this new fossil. And I'm glad you decided to share it on the show.
Yeah, definitely. I just didn't mention because I know I didn't mention it before. Yeah, the dinosaur connection is really interesting because the paper authors, they really only considered dinosaurs and mammals,
Mm-hmm.
and they ruled out the mammals by nature of their small size.
Right.
So that's really like one of their biggest, like, well, what else would be eating a palm tree?
Yeah.
The dinosaur.
Yeah,
Right?
pretty much.
So that is interesting to think about. Yeah, it's like if this may be a case that is against sauropods having beaks or beak-like structures, maybe that implies then that there's some soft tissue anatomy in their mouths
Right.
or around their mouths that we're not really considering.
Mm-hmm.
'Cause I know there's the whole thing about the hyoid in dinosaurs,
Mm-hmm.
and we have a hyoid for a few dinosaurs, like ankylosaurs, that seem to suggest that their tongues were really muscular,
Right.
or at least were mobile to an extent. So I don't know if we have anything like that for sauropod dinosaurs. Yeah, I'm glad you appreciated this paper, and I'm definitely intrigued about your second story here, our final story for the day.
Yeah.
We move to the next slide. What on earth am I looking at here?
Yeah, so let's talk about more fish weirdness in this final story. So first take a good look at this little clip here. Yeah, when I first saw this clip, that was pretty much my first thought too, was what the heck is happening? And also, this is one of the weirdest bits of anatomy I've ever seen in a vertebrate, and I have to say that's saying a lot. So, yeah, what is going on? So, first of all, this animal is a fish, and, well, the authors basically wanted to find out the answer to the same questions that we have. So, let's go on to the next slide. So, That fish is a species called the hingemouth. It is only found in freshwater environments in West Africa. Um, and it belongs to a group of fishes called the gonorhynchiformes. And the most famous member of the gonorhynchiformes is probably a marine fish called the milkfish, and that fish is a common food fish in some parts of Asia. So places like Taiwan and Indonesia and the Philippines often eat milkfish. But maybe you've never been to any of these regions and you're like, that doesn't help me at all. I had never heard of a milkfish. Well, if we go outside the gonorhynchiformes, the gonorhynchiformes belong to a bigger group of fishes called the Ostariophysi, and that's a very diverse group. That group also includes the cypriniforms, like we mentioned earlier. So Danionella is also an Ostariophysin, and so are carp and goldfish. It also includes things like the tetras and piranhas. Tetras are a very common freshwater aquarium fish, so if you keep freshwater aquariums, you're almost certainly familiar with those. And it also includes the catfishes, which are an incredibly diverse group, just a massive group. All of those fishes are members of Ostariophysi alongside the gonorhynchiformes. So it is quite interesting that both the stories I picked are not just about ray fin fishes, but they're both about Ostariophysin fishes. So that's pretty cool. Now, one of the features that characterizes the Ostariophysi is that they have a series of little bones that link their swim bladder. Oh, here comes the swim bladder again. They link the swim bladder to their inner ear. Well, fishes don't have external ears like we do, but they do have inner ears. And so the chain of little bones that links the inner ear to the swim bladder in the Ostariophycins helps them be more sensitive to sounds because they can basically hear using their swim bladder instead of just with the ear alone. And so I guess in light of that, it's not so surprising that you'd have forms like Danionella within Ostariophysi that are specialized for making sounds, right? For communication. So that's pretty neat. But when it comes to the hingemouth, definitely its weirdest feature is that whatever is going on with its face that you just saw. And so the hingemouth has this unique feeding apparatus where it has this structure that comes out of the front of its face, almost like a proboscis. So it's almost like an elephant's trunk. And so the authors of this paper wanted to find out exactly what was going on with that. And so they studied the anatomy of the skull of the hingemouth, both from observing living specimens. So that clip I just showed is a clip that is included in the paper, where they took video of what the hingemouth does when it's feeding. And that video was slowed down. So in life, if you were actually observing it, that trunk would be moving a lot faster than you saw in that video. But you wouldn't really be able to tell what was going on if it was in real time. And so they slowed it down. And in addition to that, they also looked at preserved specimens from museums to figure out what the underlying kind of skeleton and muscles were like. And so they finally came up again with a model for how their feeding apparatus actually worked. And so, where do we begin? Well, on this slide here, on the top right, you can see the skull of a quote-unquote more normal fish skull. And so, in fact, this is a type of cypriniform. So again, this is a member of the carp and Danionella group that we mentioned earlier. And so this is a more normal-looking fish skull. And so the lower jaw is marked in light blue, or the frontmost bone in the lower jaw is in light blue. It's called the dentary. And it connects to the rest of the skull by a joint, just like in our skull. The upper jaw, at least the front of the upper jaw, is formed by two bones. One is called the premaxilla, that's the one in front, and it's labeled in light yellow here. It's very small, or at least mostly hidden in a typical fish skull. And the second bone that comes right behind it is called the maxilla. It's colored in purple here. Now, in our skull, and in the skull of typical land vertebrates, so most groups other than lizards and birds, there's only one place in the skull that can actually move. And that is the jaw joint that opens and closes our jaw, right? So that's the only part of our skulls that we can move. And that is pretty normal for land vertebrates, but it's not so normal for fishes. In fishes, there are a lot more moving parts in the skull, and different groups of fishes have taken this to kind of in different directions, to different extremes. So in a typical ray-finned fish skull, such as this one, the front of the mouth can actually protrude as the jaw opens. So there are like joints that can shoot the front of the mouth forward as the lower jaw opens. So it's not just the jaw joint connecting the lower jaw to the rest of the skull that can move, but there are also joints in the kind of upper part of the skull too that can make the mouth parts do some pretty interesting things. So that's pretty weird to us, but it's not nearly as weird as what's going on in the hingemouth, which has the skull pictured on the top left here. So what's going on with the hingemouth skull? Oh, it's kind of hard to make out. But one of the weirdest things about this is that the jaw is basically turned backwards, if you can believe that. So look at where the jaw joint is. So you can still see that the makeup of the bone's the same. It's still got a dentary and a premaxilla and a maxilla. But where the dentary connects to the rest of the skull, or where the jaw joint is at the very front of the skull. It's not at the back of the mouth. So basically, the entire jaw has been turned around 180 degrees so that the jaw joint is now at the front of the skull. Weird. Okay, so how does this work? So based on the author's muscular observations, they found that there's a set of muscles that in some fishes normally opens the jaw, that probably moves the dentary as well in the hinge mouth. But of course, because the hinge mouth has its jaw reoriented like this, instead of kind of lowering the dentary downward, it flips the dentary forward instead. And so as it does so, that helps it deploy that proboscis, which is in turn attached to the upper jaw bones. And there are additional muscles that can probably help control the movements of the upper jaw bones and give the proboscis some mobility. That being said, there are no muscles that are only contained within the proboscis itself. So it is unlike an elephant's trunk in that the proboscis itself does not have that much flexibility. So it can't move the proboscis alone without moving its jaw bones basically. But with the muscles that are attached to the jaw bones, it can deploy the proboscis and protrude it forward. And as it does so, it brings the actual opening of the mouth, which is at the tip of the proboscis, in contact with whatever is below it. Now, the tip of the mouth, where the mouth actually opens, is not directly supported by bone. So in this way, it is also very different from pretty much all other vertebrates. Because pretty much all other vertebrates, the actual opening of the mouth is bordered by bone, and that's normal, right? But not in the hinge mouth. The hinge mouth instead has its mouth opening at the tip of the proboscis, and kind of surrounding the mouth opening is not bone, but instead these cartilaginous quote-unquote lips that kind of can be open and closed to some degree. And there are also very tiny keratinous projections, almost like tiny teeth that cover the tip of the proboscis as well. And so probably the movements of these structures as the proboscis protrudes helps the hinge mouth scrape off food particles on different surfaces that it's feeding on. So this might be algae, this might be bits of organic matter and detritus, this might be little insects or insect eggs that it's feeding on. Now, I mentioned earlier that in a lot of ray-finned fishes, the jaw can protrude forward as the mouth opens. And it turns out that the speed of jaw protrusion in most other ray-finned fishes is a lot faster than it is in the proboscis of the hinge mouth. So I said that video earlier was slowed down. It's actually much faster in real life. But compared to how fast a lot of other ravenfishes can shoot their jaws forward, the hingemouth still qualifies as being relatively slow. That kind of makes sense if you consider what they're feeding on, because in a lot of other ravenfishes, the main benefit of that jaw protrusion is to quickly create an area of suction. So they're using suction feeding to suddenly suck their prey items into their mouth. And without having to get very close to it, they can just kind of approach it up to a certain point, and before it can get away, they open their mouth and shoot their mouth forward and engulf the prey item. And so they need to be pretty quick to do that. Whereas the hinge mouth is mostly feeding on kind of static substances on the bottom of like the pond or the fish tank or wherever. And so it doesn't need to be so quick when it comes to protruding this very strange proboscis. So probably the reason it has its proboscis is maybe that dexterity is more important to it. So using this kind of proboscis structure, it can feed on many different types of surfaces. It can feed on wood or rock or sand, and it can manage to scrape stuff off of any of those surfaces pretty effectively. But that does raise a question. Why is it the only fish in the world that apparently has such an adaptation? In fact, there are other types of detritus feeding fishes that feed on the bottoms of rivers and lakes, and they don't have this kind of structure. And yeah, it's hard to say exactly why this specific structure evolved in the hinge mouth, but it's possible that the reason why it is the only species that has this today is that probably evolving this structure has placed a lot of constraints on how adaptable the mouth parts of a hinge mouth can be. Like, once it has been specialized into kind of deploying this proboscis, it might not really be easy for it to evolve into doing anything else. And so if the hinge mouth needed to adapt to a different type of feeding strategy, it might well not be able to because it's so specialized for doing this thing, even though it's pretty good at it. Whereas in other detritus feeding fishes with more normal fish mouth parts, it might be a lot easier for them to adapt to different types of ecologies, and so they can become a lot more diverse in their terms of their species number. But what works for the hinge mouth has worked for it so far, and it definitely gives us a lot of really interesting anatomy and evolution to look at. And so yeah, this is definitely one of the strangest bits of vertebrate anatomy that I know of, and so when I learned about this paper, I definitely had to share this because yeah, fishes are pretty darn fascinating, I have to say. Like, after reading the two papers I have for this news episode, I mean, I never disliked them, but yeah, they certainly do some really cool things that you probably wouldn't be able to come up with in your wildest dreams. So. Yeah, the hinge mouth is definitely a species I won't be forgetting anytime soon. Do you have anything to add?
Yeah, I mean, I certainly agree with you completely. I had never heard of this fish before you told me about it, in preparation for this episode,
Yeah.
and I'm really glad I learned about it now. Um, yeah, gosh, what a curious adaptation. It's like, well, I guess one of my questions is, so this is a freshwater fish,
Mm-hmm.
and how big is it?
Not that big, I think. Something you could keep in a tank.
Interesting. Okay. I just think about like how this thing would have evolved in the first
Yeah,
place, like
right.
what sort of genetic like blueprints and transitions would this fish have gone through to get to like turning its mouth parts that way.
Yeah,
It's
right.
so bizarre. And essentially like reinventing its mouth opening with
Yeah,
cartilage.
yeah.
That is fascinating. Wow. Gosh, I'm a little speechless to be honest. I just it's an impressive piece of evolutionary engineering,
Yeah.
and as specialized as it is too. Yeah, I think as far as vertebrates go, fish certainly run the gambit. I mean, they're the vertebrates that have been around the longest, and they've had the most time to come up with the most bizarre adaptations. I think it just kind of goes to show that the weirdest fish in the world aren't necessarily the ones that live the furthest down in the depths of the ocean.
Right.
Sometimes they're in a wetland in West Africa.
Yeah.
um, Which is funny because like I don't know what it is. I don't know if you've ever gotten this vibe before, but it feels like freshwater fish versus saltwater fish. There's always a bias towards the saltwater fish being the
Yeah,
most interesting.
indeed.
The freshwater fishes are the ones that we fish and we eat,
Yeah.
and that's usually the end of it. But even among the freshwater fish, there are surprises.
Hmm.
Um, I mean, especially if you're not like very familiar with fish, like I mean, just look at the cichlids in East Africa,
Yeah.
for example, or some of the fishes in the Amazon River that have adapted to the extreme mud by becoming blind.
Yeah.
Um, Remarkable. There's always something new to learn with fish, that's for sure. I'm glad you picked this paper too. I try to read widely in natural history. Um, so I have like a vague understanding of like the scope of life on the planet,
Yeah.
but beyond that, there's always something new to learn, and I'm certainly glad to have known this fish now. I kind of want to read up more on it,
Yeah.
see if I can find it in other literature, prior to this paper, to see
Hmm.
like if what people made of it back then.
Yeah.
Um, because I can't even imagine like the first, you know, scientists that stumbled across this fish, and they must have been like, did we find like a damaged fish?
Right.
No, there's more of them. They're supposed to be like this. Okay, we'll
Yeah.
go with that. That's amazing. What an amazing paper. Well, then I guess, yeah, if there's nothing else that you'd like to add, then I think this is a pretty good note to end on.
Yeah.
That's it for our news episodes today. We want to thank you all so much for tuning in and listening to us share these stories. There's always something new coming out of natural history, and if we had the time, we'd include a lot more
Hmm.
papers. But sometimes these are the most exciting ones that we could find. So let's move on now to our general acknowledgments. So on our next slide, of course, we're on Patreon, patreon.com/time and clades. So that's, of course, a website where you can donate a certain amount of funds, and those contributions will help us, continue the series, develop new projects and expansions, and just maintain things. Um, so if you're interested in that sort of thing, um, if you donate certain tiers, there are perks. You get shout outs. You can suggest topics and ask questions that we can answer on our show. There's all kinds of options available for you. Like right now, we have a couple of patrons that have tiers where they're owed shout outs. So we want to give our very specialist thanks to my sister Julie and our friends Paul, Denver, Frankish, Tristan, and Valdenunzio. So we want to thank you all so much for your contributions. It really means the world to us. And if you're interested, give it a look. Any monetary donations are accepted. Now, of course, our general acknowledgments. We want to thank our good friends Henry and Alicia for their contributions to the series. Henry, of course, is responsible for the wonderful theme music that opens each episode, and Alicia is responsible for the color scheme of Albert's Alvarezsaur avatar. Cheerful little fellow. Now, of course, we move on. We are still on Twitter at Time and Clay, where we generally will upload episodes as they air and occasionally give announcements. Though most likely you are going to be watching us on our YouTube page, that is Through Time and Clades. If you're interested in liking and subscribing our videos, it's always welcome. Get notifications. Um, if you have any questions at all about the show, whether it be the stories that we've talked about today or anything else, there are three ways to contact us. You can send us an email, timeandclades@gmail.com. You can give us a comment on our YouTube page, or you can tweet at us, and we will almost certainly get those and respond in kind. Now, if you want to check out the papers that we've talked about today, as well as any other references or things that we've talked about, you can go to the description for those links, and those will all be there for you to explore. Now, in terms of what's coming up next, this is going to be a little bit of a busy week for us because we're going to be recording 2 episodes today. So many of you are probably anticipating when the release of Humanity: A Prologue Season 2 is going to drop. Well, this Thursday, at the time of recording, we are going to record our premiere episode for that lecture series. So you're probably not going to have very long to wait until the season premiere drops.
Yep.
And I spent quite a while working on it. I'm pretty happy with it, and it's going to be a very fun introduction to what's going to be a very long and intriguing lecture series on sociocultural anthropology and its evolution through time and around the world. And I can't wait to get started on that and kind of bring some anthropology back to Through Time and Clay. It's been gone for a little bit, but it's been simmering in the oven. It's getting ready to come out and cool, and we'll share that all with everyone. So we have that to look forward to coming soon, as well as our usual news episodes that we do. Of course, March is almost on its way out, and who knows what kind of fun stories we're going to be having for our next news episode. But until then, that is it. And we want to thank you all so much for joining us, and we hope you have a wonderful day.
Yeah. Take care, everybody.